Imagine a highly guarded vault inside your body. It contains your reproductive future, sure, but it also produces, the vital hormones that protect your heart, your brain, your bones, and your metabolism.
Right.
Now, imagine the vault's own security guards, like the very cells designed to protect this precious cargo, suddenly going rogue. They lock the doors and they slowly poison the room from the inside out.
It sounds, honestly, like a thriller movie. But it is actually one of the most profound biological paradoxes in human aging.
Yeah.
We are, of course, talking about the human ovary.
Exactly.
Its function peaks in your 20s, starts a pretty steady decline in your 30s, and typically reaches complete structural and functional failure around age 50.
Right. And for generations, society and even medicine have treated this decline, menopause, basically purely as a fertility issue. Like it's just the end of your baby-making years.
Which is completely missing the bigger picture.
Exactly. But if you are listening to this, you likely know it is so much more than that. When that vault shuts down, it triggers roughly 30 years of systemic bodily changes.
Yeah.
We are looking at massive spikes in the risk for osteoporosis, cardiovascular disease, obesity, Alzheimer's disease, and diabetes.
And with global life expectancy where it is today, people experiencing menopause are spending a massive, massive portion of their lives dealing with the fallout of that single organ failing.
Right, like a third of their life.
Exactly.
Okay.
So delaying this process isn't just about, extending the window for having children. It is a critical health span issue. But the roadblock has always been that you can't delay a collapse if you don't actually know how the building is falling apart in the first place.
Which is exactly our mission for today's deep dive. We have a fascinating stack of research in front of us, anchored by this massive new study published in Nature Aging.
And the researchers behind this paper did something truly unprecedented.
They really did.
They managed to map the exact microscopic cellular mechanisms of human ovarian aging. And in the process, they might have found a real actionable pause button.
Okay, let's unpack this, because they essentially set out to draw a hyper-detailed blueprint of this biological vault as it ages.
Right, and to get pristine data, they couldn't just look at mice right away, right? They needed human tissue.
Which is hard to get.
Very hard. But they gathered 15 healthy human ovaries from volunteers who were undergoing surgery for unrelated reasons. They meticulously screened them and categorized them into 3 distinct life stages.
So they had a young group, which was ages 18 to 28, a middle age group, ages 36 to 39, and an older group, ages 47 to 49.
Exactly. But, knowing the vault breaks down is one thing. To figure out the why, they essentially had to draw a blueprint of a burning building.
Right, and how do you even map a human ovary at a microscopic cell-by-cell level? I imagine you can't just stick it under a standard microscope and count the cells.
You can't. A standard microscope shows you shapes, but it doesn't tell you what the cells are actually doing.
Right.
So to figure that out, the researchers deployed 2 incredibly powerful genetic technologies. The first one is single-cell RNA sequencing, or cRNA-seq.
Okay, I've seen cRNA-seq pop up a lot in longevity papers recently. My understanding is that it's kind of a destructive process, right?
Yeah, it's one way to put it.
Like, imagine taking a highly complex, bustling city and just dropping the entire thing into a giant blender until it's just a soup of individual components.
That's a very vivid way to picture it, but biologically, yeah, it's accurate. They take the solid tissue and literally break it down until every single cell is floating independently.
Wow.
Then they sequence the RNA of each individual cell.
Right.
Because, RNA is the messenger, right? It tells us exactly which genes that specific cell has turned on or off at that exact moment. And they analyzed an astonishing 92,965 individual cells this way.
So by blending the city, they can essentially interview every single resident and ask, what is your job?
Yes. And they categorized those 90,000 plus residents into 8 distinct types. You have the oocytes, which are the actual eggs.
Okay.
You have granulosa cells. Then you have the theca and stroma cells, smooth muscle cells, endothelial cells, and various immune cells that are circulating through, like monocytes, NK cells, and T lymphocytes.
But if you blend the city, you lose the geography. You know the baker and the mayor exist, but you don't know who lives next door to whom. You don't know the layout of the vault.
Which brings us to the second technology, which is spatial transcriptomics, or ST-seq. This technology is revolutionary because it allows us to leave the city completely intact.
Oh, nice.
Yeah, we take a microscopic slice of the tissue and we lay a high-tech barcode grid over it.
Like laying a transparent coordinate map over a photograph.
Exactly like that. When the RNA from the tissue binds to the grid, The barcode tags it with its physical location. So now we don't just know what genes are active, we have a physical color-coded GPS map showing us exactly where they are active in the tissue.
Let me just pause and make sure I'm wrapping my head around this architecture because this seems crucial. When you combine the blended map and the GPS map, what does the intact city actually look like?
It reveals a highly stratified VIP ecosystem. At the very center of the follicles, you have the oocytes, the eggs. They are the ultimate VIPs.
Right.
And wrapped intimately around them, essentially physically touching them, are the granulosa cells.
So the granulosa cells are the bodyguards.
Bodyguards, nannies, life support systems. I mean, they are entirely dedicated to keeping the VIP alive.
Yeah.
And surrounding all of that, making up the vast structural majority of the organ, are the theca and stroma cells. Think of them as the roads, the scaffolding, and the infrastructure of the city.
And as the city ages, from the 18-year-old group to the 49-year-old group, the clinical markers perfectly mirror the physical collapse.
They do.
The paper noted anti-Müllerian hormone, or AMH, which is the classic blood test doctors use to check ovarian reserve. In the young group, it was robust, sitting at 3.76 nanograms per milliliter.
Yep.
By middle age, it dropped to 1.35. And in the older group, the AMH was nearly undetectable. The city was just physically disappearing.
The structure is literally collapsing in real time.
So just to do a quick recap for everyone listening before we move on, we've got this incredible map of the ovary using cRNA-seq to see what the cells are doing and ST-seq to see where they are. And it shows this VIP ecosystem collapsing with age.
Exactly.
Which makes me want to ask you, the listener, a question. If you could generate a cell-by-cell GPS map of any organ in your body right now to see how it's aging, which one would you choose and why? It's wild to think we have the tech to do this now.
It really is. But
Yeah.
that collapse raises the million-dollar question, right? What is actually driving it?
Right, because it isn't just a passive running out of energy, is it?
No, not at all. The structural rusting of this city is actively driven by a biological process called cellular senescence.
Cellular senescence. Okay, if you follow longevity science, you hear this term everywhere. We are basically talking about zombie cells. These are cells that have taken some sort of damage, they stop dividing, but they stubbornly refuse to die.
Right. And from an evolutionary standpoint, senescence is actually an anti-cancer mechanism.
Oh, really?
Yeah, if a cell's DNA gets damaged, you don't want it dividing and creating a tumor, right? So it pulls the emergency brake and stops replicating. Normally, it would then undergo apoptosis, which is programmed cell death, and politely remove itself so the immune system can clear it out.
But zombie cells refuse to self-destruct.
They linger. And the major problem is that they aren't just sitting there quietly. Senescent cells secrete a highly toxic cocktail of proteins and chemicals known as SACPs.
SSPs.
Right. That stands for senescence-associated secretory phenotypes.
I've always thought of SACPs as toxic gossip.
Toxic gossip.
Yeah, like one worker in an office becomes a zombie cell, stops doing their job, and starts shouting toxic, stressful gossip over the cubicle walls until their neighbors get stressed, malfunction, and become zombie cells too. The damage just spreads.
That is a great analogy. You're close, but the reality is much more malicious than just gossip. SSPs aren't just annoying signals. They are potent inflammatory chemicals that physically degrade their neighbor's tissue and alter their DNA.
Wow.
And in the older ovaries, the researchers found a massive accumulation of this exact zombie cell behavior.
But how do they actually measure that? I mean, you can't just look for a tiny zombie flag.
No, you look for the trash the zombies leave behind.
Yeah.
They stained the tissues and found a huge buildup of lipofuscin.
Lipofuscin?
It's essentially biological sludge. It's a pigment that accumulates when a cell's internal recycling system breaks down and it can no longer clear out its own waste. They also found high levels of an enzyme called beta-galactosidase, which is a universal marker for senescent cells.
But reading through the study, the most alarming marker wasn't the trash. It was a specific gene called CDKN1A, or it goes by the name P21 as well, right?
Right. CDKN1A is the master brake pedal for the cell cycle. When this gene is highly expressed, it halts the cell from functioning entirely. And in the older ovaries, the expression of CDKN1A spiked dramatically across almost all the different cell types in the organ.
So the whole city is pulling the emergency brake. But what about the VIPs? What is happening to the eggs themselves while the city around them turns into a toxic, sludgy wasteland?
The oocytes are suffering severe structural damage. The researchers looked at the communication pathways, how the oocytes were talking to the surrounding cells, and they found a huge spike in a specific interaction called the MDK-LRP1 pair.
The paper called that a receptor-ligand interaction. Can you translate that for us?
Sure. Essentially, a ligand is a key and a receptor is a lock. When the MDK key binds to the LRP1 lock, it acts as a biological distress beacon.
Okay.
The specific beacon plays a crucial role in chronic inflammation because it actively pulls inflammatory immune cells, like macrophages, directly into the area.
Wait, so the aging, stressed-out egg is sending out a signal that accidentally summons an inflammatory immune response right to its own doorstep.
Exactly. And that localized inflammation heavily exacerbates the DNA damage inside the egg.
That is wild. Let's do another quick recap. Basically, the ovary ages because of cellular senescence. Zombie cells build up trash, they hit the CDKN1A brakes, and they spread toxic CSP inflammation, which damages the eggs.
That's the mechanism in a nutshell.
So, listener, think about this. Does knowing that aging is driven by active zombie cells rather than just passive wear and tear change how you view the aging process? Because for me, it makes it feel like an active battle.
It definitely is an active battle.
And if you're listening to this, you're probably wondering why evolution would allow a system with such a massive design flaw. I mean, if the VIP oocytes are suffering this much damage, my immediate thought goes to the immediate environment. What are the bodyguards doing?
Right.
Why are the granulosa cells letting these inflammatory immune cells anywhere near the vault?
And that was the exact logical leap the researchers made. They had to interrogate the granulosa cells. And this is where that spatial transcriptomics map, the GPS map we talked about, became incredibly vital.
Because it shows exactly where they are.
Exactly. Because they discovered that the bodyguards are not just one monolithic group of cells. Based on their physical location, there are 3 highly distinct subtypes.
Right. GC subtype 1, 2, and 3. Break down where they stand and what they actually do.
Okay, so GC subtype 1 is located in the cumulus. That means these cells are positioned right up against the oocyte. They are physically touching it.
The inner circle.
The absolute inner circle. Their primary job is producing massive amounts of ATP, which is cellular energy, and forming physical gap junctions to pump nutrients directly into the egg.
Wow, okay. What about the others?
Then you have GC subtype 2 sitting further out in what's called the mural layer. These cells act more like factories. They synthesize hormones and transport cholesterol. Finally, GC subtype 3 is found widespread across the follicles, mostly regulating apoptosis, the programmed cell death to keep the tissue clean.
Okay, this is where the data reveals just a brutal, tragic irony. When the researchers looked to see which of these 3 bodyguards was aging the worst, like which one had the highest burden of zombie cells, it was subtype 1.
Yes.
The inner circle. The cells physically touching the egg exhibited the absolute highest levels of cellular senescence, the highest expression of that CDKN1A brake pedal, and the most toxic cateches in the older ovaries.
The biological implications of that are just devastating. The very microenvironment designed to nourish and protect the egg becomes the primary source of the poison.
That is insane.
It perfectly explains why ovarian function doesn't just slowly fade, but often drops off a cliff. The inner circle of protection becomes actively hostile.
It's a profound structural failure. The vault guards turn on the vault.
Exactly.
So, summarizing this part for everyone, we've got 3 types of granulosa cells, and subtype 1, the closest one to the egg, becomes highly senescent and toxic with age.
Yeah, the protector becomes the poison.
Which brings up a really interesting thought for you listening. Have you ever noticed in life or in complex systems how the environment meant to protect a fragile core can sometimes become the very thing that degrades it? It's almost poetic in a dark way.
It is dark. But in biology, things don't just happen randomly. There is always an upstream cause. We know the bodyguards are turning into zombie cells. We know that the CDKN1A gene is the brake pedal causing it.
Right.
But what pushes the pedal? The researchers went hunting for the master switch, didn't they?
They did, and through some really exhaustive genetic analysis, they found a single gene acting as the master regulator. It's called FOXP1.
FOXP1.
Yeah, the paper describes FOXP1 as a transcription factor. And if I remember my basic biology, because it's a transcription factor, its entire job in life is just to scan your DNA and flip other genes on or off, right?
That is its sole purpose. It is essentially a molecular switchboard operator.
Let me try another analogy here to see if I have the mechanism right. Imagine the CDKN1A gene, that emergency brake that causes senescence, as a rowdy group of hooligans trying to get into a nightclub to completely ruin the party.
Okay, I like where this is going.
Normally, in a young, healthy ovary, FOXP1 acts as a massive, intimidating bouncer standing right at the door of the club. As long as FOXP1 is standing there, the hooligans are kept out, the CDKN1A gene is repressed, and the cell stays youthful.
That is a highly accurate way to visualize it.
Yeah.
FOXP1 binds to the promoter region of the CDKN1A gene and physically blocks it from turning on. But the study revealed a critical flaw.
Uh-oh.
Yeah, as the human ovary ages, the expression of FOXP1 drastically and naturally declines.
So the bouncer just walks away from the door.
He abandons his post. And the moment FOXP1 is gone, there is nothing repressing the CDKN1A gene. The hooligans rush in, the emergency brakes are slammed on, the cell turns into a zombie, and those toxic SSPs begin flooding the inner circle.
But the researchers didn't just, observe this in human tissue and guess that it was the cause, right? To prove causality, they actually manipulated this in live mice.
This is where the experimental design really shines. They created a highly specialized, genetically modified mouse model. They used a genetic tool called CYP19A1 Cree, which acts like microscopic scissors.
Microscopic scissors, okay.
And they programmed these scissors to snip out and delete the FOXP1 gene, but only in the granulosa cells.
Wait, so they specifically fired the bouncer just in the bodyguards, leaving the rest of the mouse totally normal?
Exactly.
The collapse
It was immediate and severe. By the time these mice were just 12 weeks old, which is very young for a mouse, like peak reproductive age, their ovaries were visibly shrunken compared to normal mice.
Wow.
They had far fewer healthy follicles and a massive accumulation of dying enterotic follicles.
The paper also noted their hormone levels completely flatlined. I mean, their serum AMH plummeted, meaning their ovarian reserve was basically wiped out prematurely. But what was happening to the DNA?
When they stained the cells of these young, genetically modified mice, they saw massive spikes in a marker called gamma H2AX.
Gamma H2AX. Can you explain what that marker actually is?
Yeah, think of gamma H2AX as a physical distress flag. That is, cell plants the exact moment both strands of its DNA are snapped in half.
Oh, wow.
It is a marker of severe double-strand DNA breaks. The absence of the FOXP1 bouncer didn't just make the cells tired, it led to catastrophic structural damage to their genetic code. And practically speaking, when these mice mated, they had significantly smaller litter sizes.
So by simply removing this one transcription factor, this one bouncer, they successfully induced premature menopause.
Yes.
It definitively proves that FOXP1 isn't just a bystander, it is the central protector of ovarian youth.
Without a doubt.
So recap time. FOXP1 is our bouncer. It protects the cells by keeping the CDKN1A breaks off. When it drops, ovarian failure accelerates. And to the listener, think about this. If FOXP1 is the bouncer for ovarian youth, what other protective master switches might be quietly fading in the background of your biology right now?
It's a sobering thought.
It really is, which immediately makes me think, if we know the FOXP1 bouncer naturally disappears as we age, is there a way to artificially prop the door closed? Can we intervene?
That is the holy grail of this research. If you understand the mechanism, you can target it. The researchers turned to a class of therapeutics called senolytics, or sometimes they're called geroprotectors.
Okay, these are the drugs specifically designed to hunt down and clear out zombie cells, right? Not just suppress them, but actually force them into apoptosis so the body can flush them out.
Correct. They essentially sweep the trash out of the city. The researchers tested 3 very well-known senolytic compounds in the lab: fisetin, dasatinib, and quercetin. They wanted to see which one could best protect the granulosa cells after the FOXP1 bouncer was removed.
And quercetin was the clear winner. Now, for those listening, quercetin isn't some experimental synthetic chemical. It's a dietary flavonoid. It's a natural compound found in red onions, apples, capers, and kale, known for its powerful anti-inflammatory properties.
Though I imagine the doses they use in the lab are vastly higher than what you get from just eating a salad.
Right, vastly higher and highly purified. But in their cellular assays, quercetin successfully cleared the senescent bodyguards and delayed the aging process. So the next logical step was to test it in vivo, right, in live mice.
Yes, but they did something very clever with the trial design here. They didn't just give it to old mice. They tested it on 2 distinct age groups to find the window of opportunity.
Right, they tested 36-week-old mice, which represents human middle age, and 48-week-old mice, which represents perimenopause, or the very end of the reproductive lifespan.
And the contrast in the outcomes between those 2 groups is perhaps the most important takeaway of the entire study.
Really?
Absolutely. When they gave quercetin to the 36-week-old middle-aged mice, it worked beautifully. It significantly reduced the mRNA levels of the CDKN1A brake pedal. It cleared out the inflammatory cytokines.
Wow.
And most importantly, it physically preserved the ovarian reserve. These mice had a higher number of healthy follicles and a significantly higher proportion of successful pregnancies compared to the untreated mice.
It saved the vault.
It did.
But what happened when they gave the exact same treatment to the 48-week-old mice?
It had zero effect. Absolutely none.
Wait, zero effect. Why? If it clears zombie cells, why wouldn't it clear them in the older mice?
Because of the structural collapse threshold. Quercetin, or any senolytic for that matter, cannot bring a dead follicle back to life. It cannot rebuild the vault once the walls have completely caved in. It can only protect and preserve the reserve that is still physically present.
Ah, I see. So once the follicle pool drops below a certain critical threshold, the biological window for this intervention permanently closes. The city is just too far gone.
Precisely. It highlights a hard biological point of no return. It proves that the dream of an anti-aging pill isn't a magic wand you can just wave after the damage is already done.
Okay, final recap of the deep dive. Quercetin acts as a senolytic that successfully preserves the vault, but only in middle age. In older age, it fails. There's a strict window.
That's the key takeaway.
So listener, does knowing there is a hard biological point of no return change how you think about preventive health versus just treating symptoms as they arrive? Because it fundamentally shifts how we have to think about medicine.
It really does. Anti-aging interventions in the ovary, and honestly likely everywhere else in the body, must be fiercely preventative. You have to intervene during the middle-age timeline.
Right.
You have to step in and clear out the zombie cells before that toxic inner circle permanently destroys the VIP eggs. We have to move from a paradigm of reactive medicine, where we wait for symptoms like hot flashes and bone density loss to arrive, to proactive preservation of the tissue while it's still viable.
And that brings us back to why this matters to you, listening right now. This deep dive into a microscopic spatial map of human ovary isn't just, an academic exercise.
No, not at all.
By understanding how the granulosa bodyguards turn toxic, how the FOXP1 bouncer fades away, and how targeted compounds like quercetin can sweep the zombie cells out of the vault, we are literally looking at the foundational blueprints for delaying menopause.
And if we can translate this biology into human therapeutics, keeping that vault open longer means buying millions of women decades of endogenous estrogen protection.
Decades.
Exactly. It's about maintaining the body's natural defense against heart disease, brittle bones, and cognitive decline. It is about vastly improving the quality of the second half of human life.
Which leaves us with a final, somewhat philosophical question to ponder. If science successfully develops a targeted therapy based on these findings, say a FOXP1 activator, or a highly optimized senolytic protocol that safely pushes menopause back by 10 or 20 years, how does that fundamentally reshape the timeline of human life?
It's a profound question. If the biological clock stops ticking so loudly in a person's 30s and 40s, how do we redraw the map of our social, psychological, and professional lives?
It's a massive paradigm shift. Because right now, that vault is quietly shutting down for millions of people. But for the first time, we aren't just blindly watching this systemic fallout. We finally have the blueprints to the building, and maybe, just maybe, the tools to keep the doors open a little longer.